So good day everyone. Today we will tackle about the delayed chromosomal and extrachromosomal inheritance. But before that, kindly read our learning target. So I can discuss the concept of delayed chromosomal, extrachromosomal inheritance, and quantitative inheritance. I can appreciate the significance of delayed chromosomal, extrachromosomal inheritance, and quantitative inheritance through sharing one's ideas. And I can create a graphic organizer summarizing all my learnings in today's discussion. So delayed chromosomal inheritance and extrachromosomal inheritance are two distinct modes of genetic inheritance that involve the transmission of genetic information outside our traditional nuclear genome. What is delayed chromosomal inheritance? So delayed chromosomal inheritance refers to the inheritance of genetic material from organelles such as mitochondria and chloroplasts, which are typically passed down from the maternal parent through the cytoplasm of the cell. One of the major examples of this is Down syndrome. We all know that Down syndrome is a genetic disorder caused by the presence of an extra copy of chromosome. This additional chromosome leads to the developmental and intellectual disabilities as well as physical characteristics associated with Down syndrome. Next is extrachromosomal inheritance. Extrachromosomal inheritance involves the inheritance of genetic elements such as plasmids or prions that are present in the cytoplasm or other cellular compartments independent of the nuclear genome. Example of this is chloroplast inheritance. We already know what is chloroplast, right? So, chloroplast is commonly found in plants. So, chloroplasts are organelles responsible for photosynthesis and have their own DNA. Certain traits, such as leaf variegatation in some plants, can be inherited through the chloroplast DNA, which is typically passed down from the maternal parent. Next is cytoplasmic particles. Refers to the various organelles and structures found within the cytoplasm of cell, including the nucleus. These cytoplasmic particles play crucial roles in the cell's function and can be inherited through cytoplasmic inheritance. One of the major examples of this is mitochondria. Mitochondria are organelles found in the cytoplasm of most eukaryotic cells. They are often referred to as the powerhouses of the cell because they are responsible for the production of energy through the process of cellular respiration. Cytoplasmic inheritance is also known as extranuclear or maternal inheritance, is a type of inheritance where genetic information is passed down through the cytoplasm of the cell rather than through the nuclear DNA. Example of this is mitochondrial inheritance. We already know that mitochondria is the powerhouses of the cell, have their own DNA which is separate from the nuclear DNA. Certain genetic disorders such as Leber's hereditary optic neuropathy, or also known as LON, are caused by mutations in the mitochondrial DNA and inherited from the mother. Chloroplasts are specialized organelles found in the cells of plants and some of other photosynthetic organisms such as algae. They are responsible for the processes of photosynthesis, which is the conversion of light energy from the sun into chemical energy in the form of glucose. Mitochondria, which are organelles found in the cytoplasm of eukaryotic cells. We already know that mitochondria are often called as the powerhouses of the cell because they are responsible for the production of energy through the process of cellular respiration. And based from my discussion earlier, mitochondria is an example of cytoplasmic particles. They are called powerhouses of the cell because they are responsible for the production of energy through the process of cellular respiration. Next is we will dive into our next topic, which is the quantitative inheritance. So quantitative inheritance is also known as polygenic inheritance, refers to the inheritance of traits that are controlled by multiple genes, each contributing a small effect to the overall expression of the trait. Multiple genes refers to the situation where a single phenotypic trait is controlled or influenced by more than one gene. This is in contrast to single gene inheritance, where a trait is determined by a single gene. Multiple genes refers to the situation where a single phenotypic trait is controlled or influenced by more than one gene. This is in contrast to single gene inheritance, where a trait is determined by a single gene. Example of this is the number of genes in polygene system. The multiple genes, polygenic traits are controlled by the combined effects of multiple genes, typically ranging from 3 to 5 genes up to hundreds of genes. Regression to the mean is a statistical phenomenon that is closely related to the concept of quantitative or polygenic inheritance. Under the regression to the mean, there are 5 examples. First is the extreme values, second is the regression effect, third is the causes, fourth is the polygenic traits, 5 is the implications. Effects of gene dominance and gene interaction. In gene dominance, in the Mendelian genetics, the concept of dominance refers to the relationship between the alleles of a single gene locus. In a heterozygous genotype, one allele may be dominant, meaning it masks the expression of the recessive allele. On quantitative traits, the effects of individual genes are usually additive rather than exhibiting strict dominance relationship. Next is the effect of genes in gene interaction. In a polygenic system, the individual genes involved can interact with each other, leading to non-additive effects on the phenotype. Epistasis is the type of gene interaction where the effect of one gene is influenced by the presence of one or more other genes. Epistatic interactions can also result in phenotypes that deviate from the expected additive effects of the individual genes. Epistasis can also lead to the complex patterns of inheritance where the phenotypic expression of a trait is not simply the sum of the individual's gene effects. Next is the effect of genes in multiplying genes. First is in gene dominance. In Mendelian genetics, dominance refers to the relationship between alleles and a single gene locus. In a quantitative traits, the effects of individual genes are usually additive, but some degree of dominance can still influence the expression. Dominance can lead to deviations from the expected additive effects of gene individuals. Lastly is the gene interaction. In a polygenic system, genes can interact with each other, leading to non-additive effects on the phenotype. Epistasis is a type of gene interaction where one gene's effect is influenced by the other genes. Epistatic interactions can result in phenotypes that deviate from the expected additive effects of individual genes. Gene interaction also contribute to the complex patterns of inheritance observed in quantitative traits. So that's all for my topic. Thank you for paying attention and God bless.